diversity in the living organism. And in this, the first we are going to take up is the bacteria, which comes under kingdom monera, and it is a prokaryote. As you know, that the prokaryotes can be differentiated from eukaryotes with regard to two distinct aspects. Number one, a distinct nucleus, and number two, all the double membrane organelles are absent in prokaryotes. So now let us take up the bacteria. Anton von Leeuwenhoek in 1683 observed in the stored rainwater tiny animalcules, and the original term which was suggested by him was Dirkens. Ehrenberg in 1892 has given the term bacteria. Pasteur proved that rotting of substances and various types of fermentation are caused by bacteria, and he gave us the technique of pasteurization. This is a typical bacterial cell. You can see the outer capsule wall inside the cell, then the bacterial genome, which is nothing but a circular, double-stranded circular molecule, and other components. Koch suggested that bacteria can cause disease, and he discovered anthrax causing bacterium, that is, anthrax disease of cattle. Bijernik established the role of symbiotic bacterium, rhizobium, in the nitrogen fixation. Here is the anthrax, and this is the rhizobium bacteria. Graham introduced procedure for staining of bacterial cells. It is known as Graham stain. Whitaker classified bacteria under the kingdom monera, of course, which includes the prokaryotes. Now, again, you can see a typical bacterial cell with cytoplasm, ribosome, plasmid, flagella, sex pili, etc. Characteristics. Bacteria is small, simplest, single-celled, prokaryotic, primitive and living organism. They are cosmopolitan, means you will find them everywhere. The bacteria have rigid cell wall composed of a substance which is referred as peptidoglycan. The bacterial cell lacks well-defined nucleus, as I told you earlier. It does not have any nuclear membrane, nucleolus, and chromosomes. Such primitive form of nucleus is referred as nucleoid. It contains a double-stranded circular molecule of DNA. Bacteria generally reproduce by vegetative, and the most common is by binary fission and by other asexual methods. In some bacteria, the conjugation type of sexual reproduction takes place. Of course, you cannot call it as true sexual reproduction. This is the binary fission, which is proceeding in a geometric progression, the most common method. The great majority of bacteria are useful to living communities as they are responsible for the decay and recycling of organic material in the soil. Many kinds of the bacteria are parasites. They are responsible for most of the infectious disease in man, animals, and plants. This is Vibrio cholerae, causing the Asiatic cholera, and that is how the bacteria is a very important component of the ecosystem. It is a decomposer, and you can see how the things are recycled in nature. Size of the bacteria. General length is from 2 to 10 mu. Smallest known bacterium is Dialister pneumonicentis, that is 0.15 to 0.3 mu. Largest known bacterium is Eupoloscemum fischlisoni, which is 0.3 mm long. Earlier, it was a Spirillum volutens, which was considered to be the largest bacteria. And here you can see the smallest one, and this is the largest bacterial cell. Different forms of bacteria. Motility. Depending upon the presence or absence of flagella, bacteria can be classified as atrichous, bacteria without flagellum. Example is Pasteurella pestis, which causes plague. Monotrichous, bacteria with one flagellum at its one end. Example is Pseudomonas. And Vibrio, that is Vibrio cholerae, you have seen it. This is atrichous without any flagella, and this is monotrichous, one flagellum from one pole. Lophotrichus, a group of flagellum at its one end. Example is a spirillum. Serpens. Amphitrichus, group of flagella at both the ends. Example is Nitrosomonas. Here is the example, Lophotrichus and Amphitrichus. Then comes the peritrichus. Flagella distributed uniformly all over the body, and the most you are familiar with this, that is the example is Escherichia coli. Pili and Fimbriae. These are hair-like structures which resemble flagella. Fimbriae helps bacteria to get attached to the plant or animal. Pili helps during the conjugation process between the two bacterial strains. Fimbriae are made up of fimbrillin protein, while pili is made up of protein pilin. Now here you can say the diagram. Cell wall. The cell wall of bacteria is made up of mucopeptides, also known as peptidoglycan or murein. Mucopeptide is a polymer made up of alternating units of N-acetylelucosamine, in short NAG, and N-acetylmuramic acid, that is NAM. The cell wall of gram-positive bacteria has a thick peptidoglycan layer and teichoic acid. In gram-negative bacteria, the peptidoglycan layer is thin, and instead of teichoic acid, it has lipopolysaccharide. And here is the diagram. You can see lipoteichoic acid. Teichoic acid is there. It is the peptidoglycan, and then here you can see the periplasmic space between the membrane and the cell wall and the plasma membrane. Slime layer or capsule. Some bacterial cells are surrounded by a viscous layer which is known as slime layer. This layer consists of polysaccharide, and you can see this is the capsule wall which helps the bacteria to thrive under unfavorable conditions. Plasma membrane. The plasma membrane is made up of proteins and lipids and is selectively permeable. That is, the plasma membrane is performing the function which it performs in eukaryotes also. The infoldings or invagination of the plasma membrane are called mesosomes, and they are associated with number of metabolic processes. Mesosomes are abundantly present in the gram-positive bacteria as compared to gram-negative bacteria. They are connected with DNA replication and septum formation in the dividing cell. And here you can see the infoldings of the plasma membrane forming the mesosomes. Cytoplasm. The cytoplasm does not show streaming movement. This is cyclosis. As you know, cyclosis you find in eukaryotes. It contains 70S ribosomes, which are usually found scattered. Membrane-bound organelles such as nucleus, chloroplast, mitochondria, endoplasmic reticulum, etc. are absent. Photosynthetic bacteria contain photosynthetic lamellae. You can see the photosynthetic bacteria. The process of photosynthesis in bacteria differs from the other plants, that is, the higher plants and few cyanobacteria. Photosynthetic bacteria may be green sulfur, purple sulfur, or purple non-sulfur. They do not use water as an electron source, hence, they do not produce oxygen in photosynthesis. You can see the green sulfur bacteria and the purple sulfur, and here you will see the hydrogen donor is H2S, so the byproduct here it will be sulfur. Nuclei. There is no distinct nucleus with a nuclear membrane. The bacterial genome is a single circular double helical DNA molecule. DNA is not bound by histones. In some bacteria, extra pieces of circular DNA are also found. These are called plasmids. You can see the nucleoid, that is the DNA of the bacterial cell. Plasmids are important in genetic recombination and in cloning of genes. You see the plasmid here, there is a conjugation tube, that is a conjugation occurring in the two strains, and at the side you can see a bacteriophage also infecting, that you will study under transduction. Plasmids which can integrate into bacterial DNA are called episomes. A. F plasmid, called as fertility or F factor or F plasmid, it plays a role in the conjugation in E. coli. B. R plasmid. This plasmid confers resistance to bacterial cell against antibiotics. Cold plasmid, it contains genes for the synthesis of colicins, that is, bacteriocins. Technique of staining the bacteria. Graham stained the bacteria with crystal violet, an iodine solution, and then he washed them with acetone or alcohol. Gram-positive bacteria becomes deep violet or purple, but gram-negative bacteria are decolorized. If gram-negative bacteria are counter-stained with safranin, they take the red color, and this is the diagrammatic representation of the gram-positive and the gram-negative bacteria. You can see the gram-positive and the gram-negative, and the cell wall, that is the peptidoglycan. Then you have the periplasmic space,